Hello everyone. Today, let's talk about why cleaning in pharmaceutical manufacturing is a science, not just a chore. Imagine a factory making heart medication one day and aspirin the next. Even a tiny trace of the wrong drug could be dangerous. This risk of cross-contamination is why cleaning validation is essential. Cleaning validation is scientific proof that our cleaning process reliably removes all traces of previous products and cleaning agents. It's not about looking clean. It's about being verifiably clean. We must protect patients from getting the wrong medicine, the wrong dose, or harmful chemicals. It's a core part of good manufacturing practices, or GMP. If residues remain, they can affect the quality, safety, and effectiveness of the next batch. A validated cleaning process ensures every batch is pure and safe. It's our safety net, structured, documented evidence that our cleaning works every time. Regulators require it, but more importantly, it's our ethical responsibility. Cleaning validation is how we prove we're in control and committed to patient safety. It's not just a regulatory hurdle, it's science in action. That's why cleaning validation is a big deal in making medicine. Now, let's look at what the regulators expect. Agencies like the FDA, EMA, and PHS all require formal cleaning validation programs. You can't just claim your equipment is clean, you must prove it with data. Written procedures, validation protocols, and scientific justification are mandatory. The FDA's regulations demand documented cleaning and maintenance to prevent contamination. The EMA goes further, requiring health-based exposure limits using toxicology data to set safe residue levels. Regulators want a risk-based, science-driven approach focused on patient safety. International guidelines like ICH Q7 and Q9 emphasize quality risk management. The message is clear: your cleaning process must be controlled, validated, and thoroughly documented. Regulators worldwide are aligned on this. Cleaning validation isn't optional, it's essential for compliance and patient protection. So, how do you build a cleaning validation program? Start with a cleaning validation master plan, your blueprint for strategy, equipment, products, and cleaning agents. Use risk assessment to focus on the highest risks, like the hardest to clean or most potent products. Group similar products and validate the worst-case scenario to cover the rest efficiently. Define clear responsibilities for every team member involved. Quality assurance, manufacturing, engineering, and the lab must all work together. The master plan ensures everyone knows their role and the process runs smoothly. Prioritize science and logic over unnecessary effort. Teamwork and communication are key. A strong plan is the foundation of effective cleaning validation. The big question: how clean is clean enough? We need measurable acceptance limits for residues, based on science, safety, and regulations. The health-based limit uses toxicology data to set a safe daily exposure, called the PDE. From the PDE, we calculate the maximum allowable carryover into the next product. Another method is the 10 parts per million, ppm rule. No more than 10 ppm of residue in the next batch. The visual limit is also required. Equipment must look clean to the naked eye. But visual checks aren't enough. Many drugs are invisible at unsafe levels. Regulators expect us to use health-based and ppm calculations, not just visual inspection. We always back up visual checks with sensitive analytical testing. The strictest limit, health-based, ppm, or visual, becomes our acceptance criterion. This ensures patient safety is always the top priority. Setting and justifying these limits is a core part of cleaning validation. That's how we define clean enough. Let's see how these limits are calculated. Suppose drug A's PDE is 0.1 mg day, and drug B's batch is 100,000 grams with a 10-gram daily dose. The health-based formula MAC =, PDE of AX batch size of B, largest daily dose of B. Here, MAC =, 0.1 milligrams x 100,000 G, 10 G =, 1,000 milligrams. For the 10 ppm rule, MAC =, 10 ppm x batch size of B in kg. That's 10 x 100 kilograms =, 1,000 milligrams. Sometimes the numbers match, sometimes they don't. Next, convert the total MAC to a per swab limit, MAC total surface area, x swab area. If the equipment has 200,000 square centimeters of surface and you swab 25 square centimeters, the limit per swab is 1,000 milligrams. 200,000 times 25 equals 0. 125 milligrams. This is the number the lab will look for. Always double-check your units and calculations. Calculating and applying these limits is essential for compliance. The process is straightforward but critical. That's how we turn science into safe manufacturing. What if your health-based and ppm limits are different? Always choose the strictest, lowest limit. Let's say product X's PDE is 0.02 mg day, and product Y's batch is 50,000 grams with a 20-gram daily dose. Health-based MAC, 0.02 milligrams x 50,000 g, 20 g =, 50 milligrams. 10 ppm, MAC, 10 x 50 kilograms =, 500 milligrams. The health-based limit, 50 milligrams, is stricter than the ppm limit, 500 milligrams. The visual limit is always required, but it's usually less strict. We must use the lowest calculated limit, here 50 mg, as our acceptance criterion. This ensures maximum patient safety. Using a higher limit would be unsafe and non-compliant. Calculating multiple limits and picking the lowest is a cornerstone of cleaning validation. It's how we defend our process to regulators and protect patients. Always err on the side of caution. That's the golden rule. Once we have our limit, we need to prove the equipment meets it. We sample the hardest to clean locations, like elbows, valves, and agitator blades, using risk assessment. The main sampling methods are swabbing a defined area or rinsing with a measured solvent. Swabbing targets specific high-risk spots. Rinsing covers large or hard-to-reach areas. Samples go to the lab for analysis. High-performance liquid chromatography, HPLC, is the gold standard for detecting specific drug residues. HPLC is highly sensitive and specific, often detecting parts per billion. Total organic carbon, TOC analysis, measures overall organic residue, useful for general cleanliness. TOC is fast and sensitive but not specific to a single drug. The chosen method must be validated for accuracy and sensitivity. Analytical results are compared to our acceptance limits. Only if all results are below the limit is the cleaning process validated. Smart sampling and analysis are the proof our process works. In pharmaceuticals, if it isn't documented, it didn't happen. Every step of cleaning validation must be clearly recorded. The validation protocol outlines the plan, objectives, scope, procedures, sampling, and acceptance criteria. After the study, all raw data and results are compiled in a validation report. The report summarizes findings, compares results to limits, and states if the process passed or failed. All documentation must be traceable and verifiable. The package, master plan, risk assessments, protocols, data, and reports proves control and compliance. Good documentation is your best defense in an audit. It's the backbone of a successful cleaning validation program. Cleaning validation isn't a one-time event. It's ongoing. Any change in products, equipment, or cleaning agents can impact your validated process. A robust change control system is essential to assess and address these impacts. New products or changes may require new risk assessments or validation studies. Even small changes like a new detergent supplier must be evaluated. Regular reviews every 2 or 3 years keep your program current and effective. Review records, deviations, monitoring data, and regulatory updates. Continuous improvement is the goal. Use data to make processes safer and more efficient. Treat cleaning validation as a cycle plan: do, check, act. This ensures compliance and a culture of quality. Ultimately, it's about protecting patients and delivering safe medicine.